Alrighty, let's gotta share the screen, share entire screen. Here we go. Okay, so today we're going to be covering this paper here. It's SARS-CoV-2 targets mitochondria exacerbating COVID-19 pneumonia. So I thought this was interesting, and this is one of the ones that got voted on. So I was like, well, what's the connection? Like, that was what kind of interested me, is like, what's the connection between mitochondria and pneumonia? Because mitochondria is going to be related to like the energy production of the cells. Like, how is that related? I don't know. Haven't read it yet. We're gonna find out together. So this is the initial chart, and I thought this was kind of interesting. I really love when papers have a figure right at the beginning in the abstract. I think it's very beneficial for this type of stuff. But there's some things that like I wanted to review and then also like look at. So this is showing phospholipid bilayer. So remember, like biology classes, you have like the polar head and then the non-polar tail, which is on the inside. And then the little more advanced stuff once you get to college is there's a negative charge of the polar head. This is the water loving side. Then you have which is hydrophilic. Then you have a water fearing side, hydrophobic, which is the internal part. So you have two sets of and it's a phospholipid bilayer. So you have essentially the reverse of two different things, sort of connected by the tail. You have a long chain of these, and this creates the membrane surrounding of the outer shell of a cell within your body. So this is a plasma membrane. It's labeled right here. And you have different types of gates that allow for things to happen. You have a voltage gated potassium channel, you have calcium channels, and then you have natural, like essentially diffusion through the phospholipid bilayer. And then you have sometimes viruses can attach to certain receptors, and I can't remember the exact phrase. We'll have to look it up. I have a couple things pulled up, and this is something we'll have to pull up too. It's like either like endocytosis or endophage or something, but it essentially just means that the reaction with the receptor causes deformation of the membrane, which allows it to then bypass and pop through. Is something that can happen, and that's how like viruses like COVID can then insert their genetic material, the RNA, inside. The lipid bilayer. So part of the reason why we have for our COVID vaccines, they have those essentially those fat molecules that sort of surround the mRNA component of the vaccine. The reason they do that is because these can just diffuse straight through the membrane of a cell. As part of the reason, and then they can be delivered straight into the body, where then it ends up getting eaten up, releases the internal contents, and then you're able to get the mRNA to get where it needs to go to be able to produce those pre-fusion spike proteins so that you can develop an immune response to those. But yeah, so this is like a blast from the past from my college time. So I think it would be good for me and like for everyone else as well to sort of like go through these different types of ways that things can pass through membranes, because I think it's going to be really relevant here. So I sort of put everything here. So first, that voltage, the voltage gated potassium channel. So why do we have these membranes on our cells? Well, it's to allow different types of gradients. So we call it an electrochemical gradient. You can have different chemical reactions that are occurring inside the cell versus outside the cell. You want to keep these separate so that you can have different things happening at the same time. Also, you want to have different types of electrical charges inside the cell versus outside the cell. But you want to be able to have some sort of balance. And how do you get this balance? Well, these different types of channels allow for this. So you can have, like, if you start to get too high of an electrical charge within the cell itself. So this bottom will say that's inside the cell. The top will say is outside the cell for this drawing right here, because that's what it is. If the charge gets too high, then the body will send an electrical signal through ATP. ATP will trigger this. That's going to be like first, the voltage is going to be like you get too high of a positive charge within the cell, too many potassium ions. So then ATP is going to come attached to this and then allow this to open because normally it's closed. So the, well, hold on. Let me double check. I don't want to speak ahead of myself because it's been a very long time. So we have closed, open. Let me just double check if it's voltage from ATP or voltage just from the charge. Yeah, so it's just from the charge itself. So what happens is you can get too many potassium ions on here. So that's going to have too much of a positive charge inside your cell. So to regain that balance, the when you have too high of a positive charge, this will actually trigger this to go from a closed state to an open state. So closed to open. And then this will allow potassium ions to flow out. Um, that's one form of gate that can occur within a plasma membrane of a cell. Then we have the calcium channels, which I think are the ones that I was confusing with. These are the ones where you need ATP, you need energy to be able to open this type of channel. So normally it's in a closed state again, and then you have ATP, um, uh, the energy signaling from the mitochondria release that's able to bind to it and then allow it an open state to allow calcium to flow in. So this would be when you have too little charge within the cell. So you want to increase that charge, and that allows calcium to come in to help balance it out. Let me just double check as well. Do, 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 do. Okay, so little off. So maybe the okay, so I'm thinking of the other type. So there's ATP driven pumps, which is going to be different. And then this is the endocytosis that I was talking. That's the word I was trying to think of earlier. I might have said it correctly. I don't remember. But endocytosis is when something binds and then is able to break through the membrane, and the membrane seals right back up. But so this is a different type. So this is another voltage type channel for. This is why it's good to refresh ourselves. So there are two. You have potassium and calcium that can flow in and out of the cell, and you need to have this in regulation. So when you have potassium, too much potassium, so the charge is too high, um, you can have the voltage gated potassium channel that's going to be triggered through these electrical signals to open up, and that's going to allow it to exit the cell to reduce the charge internally. And then similarly, if you have too much of a positive or too little of a positive charge on the inside, you can get flooded by calcium. And this looks like it can do two different types of ways for the calcium. So one, you can have a secondary reaction. So a specific type of molecule binds to the calcium ion channel in here, which forces it into an open state, which allows calcium to flood in. So that's one type of way. But then the second type of way is similar to the potassium gated channel. when there's too low of a charge on the inside, then it'll go to that open state and then allow for calcium to flood into the cell to restore the balance. And hypoxia increases well, that doesn't matter for now. So then we have the two other types. So we're going to have, and then there's also the ATP generated one, which is going to be the what I was saying before. We have an energy molecule that opens a channel that allows a certain type of action. So this is going to be more for different types of molecules, I think, that are brought into the cell for certain types of chemical reactions. So endocytosis, going to double check. Endocytosis for virus entry. Yeah. So endocytosis is what this is happening right now for the COVID. So when COVID binds to a receptor, endocytosis allows it to then create a reaction where it will sort of get engulfed by the membrane and then get spit out the other side. And then the membrane seals back up, typically. This is similar to that one paper we reviewed a long time ago where it's talking about how this sort of thing changes the shape of the cell, which makes it more prone to lysing, but which is a cellular explosion essentially. But this is how the virus enters the cell, and then what are the lipids used in mRNA COVID? And then the lipid nanoparticles is the other type, which is going to be passive diffusion through the membrane. Oh, okay. So they actually do enter through endocytosis as well. So that's a good catch on myself. So they're also being brought in through endocytosis. They're engulfed into a vesicle, which is like a transport molecule, and brought in to the cell that way. Okay. And then passive diffusion of fats into the phospholipid bilayer. So I was making a cross comparison, which was inaccurate for the lipid nanoparticles. But what can happen is that any types of fats, so these are going to be, so the phospholipid bilayer is essentially, it's like long chain fatty acids in a way. But they're not really, they're its own unique thing. But because of that, fats can easily cross through the membrane by simple diffusion. Which means that something like this. So we have like these uncharged particles or these fats that can then just passively diffuse through the cell. So they don't need a channel to be able to pass in and out. These are things that can happen as well. So now that we've sort of gotten all caught up, and I've recalibrated myself since this has been a while since I've taken these types of biology classes, we can better understand this stuff. So when we're talking about mitochondria, this is suggesting that once the RNA from COVID enters the cell. It then is attached to the ribosome, which produces viral proteins like the spike protein, but also nucleocapsid, like basically the entire COVID virus while that's being produced. These viral proteins will then interact with the mitochondrial proteins. So this is then showing the membranes of the mitochondria. And then what's happening. Oh, so there's two sets of things happening. So one, it's interacting with the viral proteins from COVID, the SARS-CoV-2 that's being produced inside your cells, because essentially what happens is it hijacks your cells to produce more viral viruses. It of itself, these viral proteins will then interact with the mitochondria. They'll also move into the nucleus of the cell. When the nucleus of the cell, they'll alter gene transcription, the ability of your cell to like produce functioning components of whatever it needs to produce, whatever it's set out to produce, um, which increases early cell death. So it's impairing the process. So the more impaired process of transcription, which is this process of creating new proteins, if that's impaired in any way, you're going to have cells that can die easier, that aren't going to function appropriately. And your body might see these like impaired proteins that are produced and see it as something that needs to be like destroyed. So it can promote inflammation. Um, and then also when it's it's increasing mitochondrial fission genes. So this fission is breaking up. So I'm guessing this is meaning that any mitochondria that is being produced through this process, when it's creating more mitochondria in your cells, um, it's resulting in protein structures that split from the mitochondria. So like right here, I think this is what it's showing. So you have a mitochondria. It's essentially doing that shape deformation thing that we saw earlier in a different paper for the mitochondria. But what's happening is that it's crumpling on itself and then splitting into two fractured mitochondria instead of a single full mitochondria like this. It's being broken into tiny pieces, which could still function, but probably don't function as efficiently because they're not the size that they're supposed to be. And what does this mean if we have impaired mitochondria? Well, that means we're going to have lower rates of energy production. We're not going to be as efficient as doing oxidative phosphorylation, I think. Oxidative phosphorylation. Let me double check. Phosphorylation, which is the energy production mechanism that allows you to actually have energy to create any types of cellular processes, which is gonna, I don't want to say like make you tired at the cellular level, but you're not going to have enough energy to actually have appropriate cellular functions, which means that you're not going to be able to like repair damage from injuries. That's going to take longer. Anything that happens that causes injury to you is going to take longer to heal, is going to more likely get worse. If you have a rapid need for an immune response, your body's not going to be able to function appropriately to mount that response. Really, everything gets disrupted this way. And then you could also like feel tired and stuff like that, cause disruption. If you don't have energy, you really can't sustain life in an appropriate manner. Yeah, so that seems like to be the whole everything from this figure, but we're gonna learn more in depth here. So sorry about that. It took 20 minutes already just talking about how things work in basic biology or a little more advanced biology too. But yeah, sorry, that's been a while, so I need a little refresher. So following entry into airway epithelial cells, SARS-CoV, and they're gonna abbreviate that AECs. Hate acronyms personally, but It is something we do in science. SARS-CoV-2 releases its single-stranded RNA into the cytoplasm, where it is translated into viral proteins. Several of these viral proteins localize to mitochondria, interact with key mitochondrial components. These viral host proteins interactions induce profound mitochondrial dysfunction, characterized by disrupted mitochondrial metabolism, altered mitochondrial dynamics, um, airway epithelial cell apoptosis, so cell death, and impaired hypoxic pulmonary vasoconstriction. So hypoxia, typically you don't have enough oxygen for something, so it's a high CO2 environment. Vasoconstriction is when the blood vessels get smaller. Constriction is getting tighter, kind of like a boa constrictor, tight it. And then so when you have ves- so if we're thinking about like the capillaries in your airway, if these are getting too small, you're not able to have enough oxygen transport. You have less room for like blood cells to go through. Maybe if it's getting tight enough, you can't even pass through certain capillaries, but I doubt that would happen. But you'd have essentially less blood cells that could pass through. You need the blood cells to pass through to be able to do the carbon oxygen exchange at appropriate levels. So this is going to slow down this process, which is going to increase the amount of dissolved carbon dioxide within your bloodstream, which can then lead to other problems. Um, but let's double check uh, this. Yep, to low oxygen levels. Yeah, that's everything I said. Um, I just want to know specifically, why is cellular hypoxia bad? Specifically what that is going to do. So in cellular hypoxia, it rapidly starves cells of oxygen required to produce energy, which acts as fuel. So okay, so that's the component that I was missing. So I told you already, what happens sort of like the macro scale. We're still talking small scale, but at the that's like the large scale in this compared to cells. So when the cells don't have enough oxygen coming in, so oxidative phosphorylation, oxygen is an important part of oxygen. Here, we have a computer. I can show you oxidative phosphorylation. Phosphorylation. I just want to have something that has the figures to show you. Here we go. So in the mitochondria, oxygen comes in, um, goes through a cycle, releases carbon dioxide, and then through this interaction, you produce energy or ATP in the mitochondria. Um, there's an important role that happens with the endoplasmic reticulum and everything like that. But at the high level, this is what's happening. So if we have lower levels of oxygen, we're going to have lower levels of ATP produced, because this is the important part in this process to actually produce energy. So we have less energy production by having less. When this vasoconstriction, which narrows the blood vessels, we don't get as much. So this is going to increase carbon dioxide levels in our blood, which is going to slow down this, and it can actually trigger a secondary process. Um, I forget the name of it. What is the other energy process outside of oxidative phosphorylation in humans? I think it's anaerobic respiration. So let me double check. Anaerobic respiration with high CO2. That's not right. I'm gonna ignore that part for now. It'll probably come up in the paper. But so we have a couple different things. So one, we have this less oxygen that's going to produce less ATP in our bodies, but also we're going to have this impairment through COVID interacting with the mitochondria to reduce the energy output as well. So we're sort of having these compounding factors over time. And this is why it's so bad. It's sort of like this, what do they call that? When a snowball rolls down a hill, these building compounding factors that can cause bad things down the line. SARS-CoV-2 inhibits HPV, likely HPV hypoxic pulmonary vasoconstriction through the impairment of oxygen sensing of electron transport chain complex 1, thereby promoting intrapulmonary shunting and silent hypoxemia. Mitochondrial damage is a conserved feature of coronavirus infection occurring with humans and coronavirus, so mouse. Coronaviruses damage mitochondria and airway epithelial cells. So these are the cells that are lining our lung tissue, pulmonary artery, smooth muscle cells, pulmonary artery endothelial cells, immune cells, and cardiomyocytes by causing rapid transcriptomic changes in nuclear encoded genes, regulating mitochondria and by viral proteins interacting with host mitochondria proteins. So that's sort of what I already told you. Coronavirus infection causes mitochondrial depolarization, mitochondria transition pore opening, inhibition of the electron transport chain and ATP synthetic apparatus, increased mitochondrial fission, apoptosis, and impaired mitochondrial oxygen sensing. So I think I'm going to have to break this down differently than what I normally do, because I'm probably not going to be able to read line by line because I think this is going to get like too complex. So what I think I'm going to do is skim through this, because I think a lot of what we already covered is sort of like the explanation here is that when COVID binds to the receptors, endocytosis occurs, it's able to pull into the cell, inject the RNA. The RNA is then translated once it's brought over to our cellular machinery to produce that through the ribosomes. And then this produces spike proteins. Some of these spike proteins and like COVID viral proteins that are produced through this can interact both in the nucleus as well as the mitochondria. When it interacts with the nucleus, it damages how mitochondria are produced. So this is one way. So this is going to reduce the production of mitochondria. So we're going to have less ability to create energy through this way. Mitochondria also are more likely to fragment and break apart into smaller components, which either will completely eliminate their ability to assist in energy production, or it'll impair their ability, so they'll be less productive. So if they're like can do normally like 3x level, now they'll be down to like a 0.5x level type of thing. I'm just making up numbers there, don't quote those. And then we have earlier cell death, because when you have things that don't look right, your body will be like, I don't really recognize you, and I'll attack it. So this can cause inflammation and also earlier cell death than what's programmed, as well as like the end caps. So you have like start and end caps of things. So if things are produced, they have like a certain lifespan, and if these end caps are not produced appropriately, you'll have a much shorter lifespan of that cell than what should happen. And then also through this process, because it's increasing the calcium concentration within the cell through this process, I'll have to like look through to see what's actually causing that specifically. This is going to create hypoxia of the cell, which is going to then result in vasoconstriction, so shrinking of the blood vessels within the lung. it's going to cause less oxygen to be able to be exchanged. So you're going to have increased levels of CO2. This creates a different. I really have to look and see what it causes. There's like a different cellular process when there's too much CO2 for energy production. I forget what it's called, um, but we'll ignore that for now. And then when you have less oxygen, you already have this like damaged mitochondria, less functional mitochondria, and then you have less oxygen. You're going to have less ability through oxidative phosphorylation, this energy production method, to actually create energy. So this is sort of like a compounded energy loss effect for us at the cellular level, which means we're not going to have ability. So there's damage to these lung structures, which allow for the gas exchange to occur. Endothelial injuries, these are the blood vessel linings, the endothelial tissue, and then subsequent thrombosis, so the blood clotting that can occur, and a severe inflammatory response. So this is going to be swelling of cellular tissue, a lot of like T cells, B cells, etc., that are going to come in, swarm in the area, and then like attack things and kill cells. Hopefully, just the virus, but can also attack other things that have been compromised or that have been damaged from the virus. And then this is like the heart connection, because the lungs feed into the heart feeds into the lungs. That's the whole exchange cycle. The cardiomyocytes are the cells of the heart. So it can also directly infect those, but we also know that it spreads throughout the rest of the body and infects other things as well. So coronaviruses disrupt mitochondrial function in disease-relevant lung cells, both by causing transcriptomic changes, the ability to read and then produce proteins within our cells, through direct interaction between viral proteins and like the ribosomes and the host mitochondrial proteins. The resulting mitochondrial damage impairs cellular energetics, the ability to produce energy, disrupts oxygen sensing, the ability to sense oxygen, and also the amount of oxygen that's normally received through the restriction of the capillaries. And then promotes cell death through induction of mitochondrial fission and apoptosis, so causing the mitochondria to get small, split, and then they can die from this. They can be killed through this process. Despite growing evidence that mitochondrial damage contributes to the pathophysiology of COVID-19 pneumonia, the central role of mitochondria in this disease remains underappreciated. So people just aren't really recognizing this as the important facet of COVID that it is. SARS-CoV-2 disrupts multiple interconnected mitochondrial pathways that are required to maintain cellular homeostasis, the ability to maintain balance within your cell. At the early stage of infection, transcriptomic analysis indicates that SARS-CoV-2 infection induces broad reprogramming of mitochondria-relevant genes in the pulmonary vascular cells, vascular veins, including PAECs and PASMCs, pulmonary artery smooth muscle cells, and then pulmonary artery endothelial cells. So these are the ones that arteries take the blood away from the heart, veins bring it to the heart. The consequence of this, and then oppositely, then arteries would bring blood to the lungs, and then veins would bring it to the heart from the lungs. The consequences of this virally triggered transcriptomic change include suppression of electron transport activity in mitochondria ATP generation, activation of mitochondrial fission, and upregulation of mitochondria apoptosis pathway. So this is what we talked about earlier. And then also there's direct COVID-19 mitochondria interactions, which cause damage as well. The profound mitochondrial dysfunction caused by SARS-CoV-2 infection, which is replicated by transfection of disease-relevant cells with individual SARS-CoV-2 proteins, is termed mitochondriopathy. So they coined a new term, which means they invented a new word. for this process of SARS-CoV-2 infection impacting the mitochondria. They're calling it mitochondriopathy. Apathy is usually just like a disease, so it's a disease of the mitochondria, is essentially what they're saying. The consequences of coronavirus infection are conserved among various types of human and murine mouse coronaviruses and include impairment of metabolism, the ability to break things down, and then ATP synthesis, the ability to produce energy. For example, SARS-CoV-2 infection suppresses oxidative metabolism, disrupts lipid metabolism, and in the longer term reduces cellular energy production in long COVID. So they're directly relating this to why we have long COVID issues, which could also play in, perhaps into ME/CFS, because the greater theory that I've been hearing is that ME/CFS is directly caused by any number of viruses. So viruses like COVID, but it seems COVID more predominantly does this than other viruses, but viruses like COVID can cause this type of issue, which then results in low energy production over the long term. So low energy, like the ME/CFS type thing, myalgic encephalomyelitis and chronic fatigue syndrome. So this could be playing a direct role in that, and this could be like the theory behind this high levels of fatigue and loss of energy, poor sleep, and all sorts of other things. Moreover, SARS-CoV-2 infection alters mitochondrial dynamics to favor mitochondrial fission, the breaking apart of the mitochondria into smaller components. This relates to viral transcriptionally upregulating dynamin-related protein, blah, 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 blah. Part of this is that coronaviruses suppress expression of mitochondria fusion proteins and increase oxidative stress. So I guess there's fusion proteins that need to be produced to be able to have the mitochondria to form appropriately. So this is how, by disrupting this ability to bind appropriately, it's promoting improper creation development, which is how it's then fissuring, breaking apart. And then another hallmark of acute SARS-CoV-2 infection is the ability of the virus to impair hypoxic pulmonary vasoconstriction, which is what we talked about before, which is a physiological mechanism that constricts small precapillary pulmonary arteries. So that's the vessels that lead into the capillaries and perfuse hypoxic alveolar segments, diverting blood to better ventilated lung regions. So I guess what happens, since I'm not like an expert in lungs, is that the arteries, so this is blood coming from the heart to the lungs, they constrict into the capillaries. So it seems like the capillaries are not as affected as I initially assumed, but the tiny arterial blood vessels that are leading into the capillaries get smaller, which will still have the same end result as what I explained earlier, where you're going to get less exchange, but also higher pressure. So what's happening is that instead of going through these normal capillary channels, it's diverting blood to better ventilated lung regions. So if you have, like, you think of your lung as a whole, they're going to be regions where it's going to have higher and lower oxygen. So like if you like breathe in, diaphragm pulls, you're probably going to have more oxygen in like the upper region than the lower region, just based off of the airflow. So like the oxygen gets pulled in, and then it's going to go like higher. You're going to have like lots of oxygen, lower oxygen down to like the lower levels, mostly because we don't probably take as deep breaths as we should, but that's essentially how I imagine it would be happening. So it's redirecting the blood flow. So your body, to compensate for this, since we're not like getting the appropriate amount of oxygen that we need, it's diverting it to higher oxygenated levels. So probably this upper lung region, and the lower lung region is not getting as much. So it's diverting things that way. to try and get more oxygen out of it, even though we're not like having as much exchange um, to make up for this deficit. Failure of hypoxic pulmonary vasoconstriction is mediated by impaired mitochondrial oxygen sensing and contributes to intrapulmonary shunting and a profound hypoxemia and a COVID-19 pneumonia. So I think what this is saying is that, let me just look up this phrase, intrapulmonary shunting. So shunting is usually like blocking off. Yeah, so it's blocking off blood flow to that area. And since it's being blocked off, your cells that would have, like, I guess in the lower chamber is my assumption, since it's blocking off the those channels from getting that, the blood will still circulate, so it's not releasing the carbon dioxide and picking up new oxygen. It's just cycling back into your body um, without being oxygenated. And the problem is that when you have something like COVID pneumonia, when you're already having difficulty and some blockages from being able to breathe, if your blood is then like diverting to higher areas to pick up oxygen and you're recycling the lower areas because the blood is still going to those regions, your body's just saying, like, we're not going to get as much oxygen we need. We need oxygen up here. So we're going to focus up here. That's going to still be returned. So you're not changing like the amount of blood flow to the lower regions, even though it's turning it off. So this is going to make it so that you're going to systematically over time have lower and lower and lower levels of oxygen, which can then have if the pneumonia doesn't clear within a certain amount of time, it's going to make your condition become worse and worse and worse and worse. And they're saying that an interesting feature of COVID-19 is that some patients with profound hypoxemia, basically high levels of carbon dioxide in the blood, not enough oxygen is getting in, have minimal dyspnea, so minimal trouble breathing. So they're not having trouble breathing, but they're still not getting enough. So like this is the interesting part of COVID, where you're having multiple things happening at the same time. They're calling this silent hypoxemia. This condition. So it's not necessarily that you're like there can be like the clotting off, the plugging off, the shunting of the lobes due to like immune reaction buildup for the pneumonia, which is typically what happens, I think. But because of like these mitochondrial issues, it's like being more profound, and this vasoconstriction is being more profound. So you can still have the ability to breathe, but you're not getting as much oxygen as you should because of all these other internal factors that are happening. So this review highlights mitochondria as a major target of SARS-CoV-2 infection and a critical nexus, a critical point feature through which the virus drives metabolic failure. So mitochondriopathy, disease of the mitochondria, through this COVID infection, contributes to many features of COVID-19. And then some preclinical studies have supported what they've been showing. So preclinical studies suggest that inhibiting viral-induced AIF. This is why I hate acronyms. apoptosis-inducing factor, mediate apoptosis or enhancing HPV are therapeutic in a murine model. So they tested messing with these channels to try and block. So once you understand an issue, you can try and have targeted therapies to prevent the issue from occurring or to like mitigate the effects of it. So they found that targeting these two different pathways had therapeutic effects, beneficial effects in mice models. But that was with in a mice model of COVID-19. So interesting, they're saying that infection with intact virus is not required to induce mitochondrial injury. Simply putting cells in contact with SARS-CoV-2 proteins that are delivered by a lentivirus also suppressed oxidative metabolism in humans. Like in human cells, sorry. So these mitochondrial energy production effects, these impairments can occur both with whole virus infection of COVID, in addition to individual viral proteins being present within the cell. Apologies if you hear the neighbor's leaf blowing. And then just a quick note on the other thing. So you're like, oh, well, you might think, oh, well, what about vaccines? Because like they produce spike proteins, but you got to remember that they're in a pre-fusion condition, which means that they can't bind to anything. So I think that would be very important in this, that I do not believe that pre-fusion spike proteins that are produced through a vaccine would have the same effect. I think that the spike protein that are produced within it, if there's viral fragments of that in the cell, it would need to be in a fusion condition, an open condition versus a closed condition to cause these impacts. That is my current belief. They didn't test this in the paper, as far as I can tell, and they're not really saying like what viral fragments of SARS-CoV-2 stimulate this. At least I'm not seeing that yet. But just to help ease any worries if you're thinking about that. And right here, they're also saying is that in addition to triggering cell death, coronaviruses can prevent cell cycle progression, um, where this might impair lung healing post COVID-19, which is what I was talking about. Is when you don't have energy to do processes, you aren't going to have the energy to help assist in wound healing, which is going to slow down the ability to heal wounds over time, immediately after the infection and during the infection. And yeah, so it is the viral fragments that they're talking about is the spike protein, the S1 and S2 subunits of it. I don't think I've talked about that before, so I guess we'll show that real quick if I can get it to pop up. S1 and S2 subunits, spike protein, COVID. So the S1 and S2 subunits of the spike protein are this. So you have the receptor binding domain is how it connects to things like the ACE2 receptor, the RBD. And then you have the S1 protein, which is the top protein, and then the S2, which is sort of like the branch protein. So here's another one. So S2 is like the top of it. S1 is the top of it. S2 is the bottom of it. For COVID, here's what it typically looks like. You can see it's kind of like this giant Y shape that's sort of a combination of several different protein structure units that bind together. And that's what the spike is on the spike protein. So this would need to be in a fusiform condition, something that's an open condition. So I guess that's another thing I can show too. Trying to find a good example for you guys because these are mRNA, spike protein. Thought this was going to be easier to show. So, here we go. So prefusion, so it's locked into this shape. So it's open. So when it interacts with something, it's supposed to close. So this is the open state, this is the closed state, and then this is the post-fusion state where it sort of like gets rigid so that it can sort of shoot. So it can like lock together with whatever it needs to. So in this open state here, it's showing exactly what it would show in a normal like SARS-CoV-2 molecule, but if it can't lock to anything, it can't cause a reaction. So like with the interaction with the mitochondria, in this case, like this wouldn't be able to occur because even if it like comes into contact where it could like interact and then cause something to happen, it's not going to because it's just existing. It's sort of like a locked statue that's just kind of like floating around. If it was a live viral fragment, it could still interact and then like close on it and then cause a reaction to occur. So you shouldn't worry with like the mRNA. Oh, is that going to give me like energy reduction levels and stuff like that? It's not because that's how it's designed. It's designed to be in a locked state, and that it can't really do anything. Well, it can't do anything. But the SARS-CoV-2 fragments that just come off the cell through whatever happens, like you can get cellular digestion of the virus, and this can leave fragments that we saw can cause issues. So these spike proteins, when they're fragments, can still attach and bind to things, which can then cause reactions like this with the mitochondria when they're in the cell. So this won't happen with the mRNA from a vaccine, just as a, or like the spike proteins produced from a vaccine, just as a heads up. Even though they're viral fragments, they're not active viral fragments. Ooh, my neck. It must have been putting it in a bad location. So this will be a good section to actually read. So growing evidence suggests that chronic disruption of mitochondrial function occurs in long COVID. Survivors of COVID-19 infection often report chronic symptoms such as fatigue, low exercise tolerance, neurocognitive dysfunction. So you need energy for your brain as well. So lowered ability to send signals. So brain fog. Along with expression of markers which correlate with mitochondrial dysfunction in terms of the tests, the blood tests that you could do. Patients with long COVID also exhibit reduced energy production, increased reactive oxidation species, so free oxygen radicals existing, which can then cause damage to things, which we don't want, and perturb mitochondrial biogenesis and immunometabolism. So this is going to be impaired creation of mitochondria, in addition to impaired like energy production through the oxidative phosphorylation pathway, the energy production pathway. Long COVID patients have impaired mitochondrial respiration and peripheral blood, mononuclear cells, and significantly lower, I don't know what that is, OXPHOS capacity in their skeletal muscle. Together, these observations support a deficit in mitochondrial bioenergetics, the energy production ability, where I just lost it, as a potential driver of long COVID. Long COVID shares clinical and biological features of post-infectious syndromes such as myalgic encephalomyelitis, chronic fatigue syndrome, where defective mitophagy and mitochondrial respiratory dysfunction have been reported in brain, skeletal, muscle, and vascular endothelium. So this is like what I talked about earlier. Emerging studies suggest that even after the active infection is resolved, residual viral proteins, particularly SARS-CoV-2 spike protein, may linger and continue to cause damage to the mitochondria by increasing oxidative stress and disrupting energy metabolism. So this is what we had talked about before. And there's also another paper with the immune system of the same exact effect, where the spike protein residue within our bodies is still binding to things and then causing issues, and then the immune system is triggering it, breaking it down. And then since it's not efficient at breaking down the spike protein, there's still remnants that are like left over after it goes through its initial processes for like reactivating and having to, it's like an iterative issue. So these viral proteins may trigger production of autoantibodies against mitochondrial proteins. So you can even have your immune system attack the mitochondria themselves, which causes the damage even more, or a reduction in levels of mitochondria even more within your body, which causes ongoing damage and issues, even the absence of an active infection. And then SARS-CoV-2 proteins can also induce neuroinflammation and neurodegeneration, which we've shown other papers that I've covered before, not necessarily here, but on my YouTube channel. In the cortical and hippocampal regions by disrupting endoplasmic reticulum mitochondrial calcium signaling, increasing reactive oxygen species production, and altering neuronal mitochondrial dynamics. These virus-induced mitochondrial defects collectively contribute to the cognitive decline and energy failure characteristics of long COVID. While these findings open new opportunities to therapeutically target mitochondrial health, it is also important to highlight the importance of structured energy conserving rehabilitation. So this is just saying like having your life be low energy to preserve energy, because you literally at the cellular level don't have enough to function. So don't overexert yourself is what it's saying when you have like this issue from COVID infections. And then that's talking about metabolic profiling to recognize patients with this issue. And then to investigate the mechanical basis of these mitochondrial abnormalities in acute and long COVID. Several studies have examined how SARS-CoV-2 directly perturbs host mitochondrial metabolism. Garneri et al. 2023 investigated how SARS-CoV-2 infection disrupts cellular metabolism of the host by targeting mitochondria in human and rodent models. Their data revealed that during acute phase of infection, the virus inhibits the transcription um, of nuclear encoded oxphos genes, causing oxphos inhibition and a shift towards HIF 1 alpha driven glycolysis. This shift in metabolism simultaneously activates the integrated stress response and innate immunity pathways. Viral infection also induces MIR 2392 expression, reducing expression of mitochondrial genes. Interestingly, autopsy tissues used in their study showed that mitochondrial function often recovers after the virus is cleared in the lungs, but the kidney and liver exhibited a sustained and profound downregulation of these genes. The study concluded that long-lasting metabolic impairment in vital organs may be a primary mediator of severe and acute COVID-19 pathology and the systemic manifestations seen in long COVID. So it's saying that in the lungs we can typically clear the virus and these types of issues, but in other tissues like the liver and kidney, the brain, um, maybe even like the heart tissue, some of these other places, the body's not as good as clearing the virus, so it can have long lasting impacts in those regions, those types of regions. And a few studies have tried targeting these mitochondrial pathways in coronavirus induced pneumonia. And it looks like they've had a little bit of success with that. But it's promising. They still need to do more research, but it's promising doing these targeted mitochondrial therapies. Um, and it might be a promising form of antiviral therapy. Cool. So a viral mitochondriopathy, pathology of the mitochondria, disease of the mitochondria, plays an important role in the pathophysiology, the development of a COVID infection. Earlier in the infection, SARS-CoV-2 induces a widespread transcriptomic dysregulation. It impairs the ability to create proteins effectively by hijacking it to create more of itself, while also damaging our internal ability to produce proteins for other things. And this directly dysregulates the production of mitochondria to be produced within the cell. Viral proteins also interact with the mitochondrial proteins, which leads to additional functional consequences. Multiple critical mitochondria pathways are disrupted, including the production of ATP, energy, mitochondrial functioning, and then earlier cell death, and their ability to sense and process oxygen. Sense and process oxygen. Just repeating that because I hit the microphone, you might not have heard. And these impairments contribute to some key clinical features of COVID, such as the profound hypoxemia in some patients with COVID-19. Persistent mitochondrial dysfunction may also contribute to long COVID. Both in vitro and in vivo studies demonstrate that this viral mitochondriopathy occurs in many coronaviruses, and recognition of this coronaviral mitochondriopathy, this disease of the mitochondria specifically, uh, may offer potential therapeutic opportunities to improve bringing oxygen in or stopping mitochondria from signaling cell death and killing off themselves within your body. So protecting these types of things could improve people's conditions. However, these need more preclinical testing before being introduced at scale to people, to real life humans affected with long COVID. So this is a promising area that probably will help with not only long COVID, but maybe also people that have been living with ME/CFS in the future. Seems a promising area. We'll see. They still would have to test these types of drugs, these targeted therapies on humans in clinical trials. So maybe in the next 3 to 5 years, we could see something where we have a treatment for this type of issue. So yeah, sorry this was a long one. I had to refresh myself because it's been a long time since had uh, uh, I looked at cell biology stuff. But I thought this was a fun one. I learned a lot, and I hope you guys did too. All right, have a good rest of your day.